So we're looking forward to these Disruptive Dozens, and our first presenter will be Dr. Luc Vanderberg, who will introduce the next one.
We'll begin the introduction.
New gene and cell therapies to fight a formidable brain cancer.
Modern medicine lacks effective treatments for glioblastoma, the most common and deadly type of brain cancer in adults. Unfortunately, most patients die within a year to 18 months of diagnosis. Now, scientists are bringing the tools of gene and cell therapy to bear on this formidable cancer. One approach involves cancer-killing viruses engineered in the laboratory to seek and destroy tumors. After the virus enters dividing cancer cells, it copies itself many times over, while leaving normal cells unharmed. As the cancer cell dies, viruses are released and infect and kill nearby tumor cells. Clinical trials are now underway to test this form of gene therapy in glioblastoma patients. Researchers are also applying CAR T-cell technology, a groundbreaking form of gene and cell therapy in which a patient's own immune cells are isolated, molecularly rewired with therapeutic powers, and then put back in the patient's body. CAR T therapies are currently used to treat certain blood cancers, but scientists are now tinkering with ways to make the immune cells multifunctional, a critical capability for fighting solid tumors, like glioblastoma, which are heterogeneous and can evade drugs directed at a single target. Another novel cell therapy could one day help glioblastoma patients. It stems from a remarkable decade-old discovery. Cancer cells that spread within the body can find their way back to their original tumor. This rehoming is spurring efforts to use CRISPR gene editing to genetically engineer patients' own tumor cells, endowing them with dual cancer-killing and immune-stimulating properties. Once the cells are placed back into the body, they can return home and destroy their counterparts and impart long-term immunity against cancer.
And we have here Dr. Khalid Shah from Brigham and Women's Institute. Khalid, you've been invested in this domain for a very long time. What are some of the remaining hurdles to bring these types of therapies successfully to the clinic?
Thank you, Luke. There are a number of bottlenecks and choices to be made, I believe. I'll start with a critical one that forms the basis of bench-to-bedside translation, and that is the preclinical studies. I think it's essential to use mouse models, particularly that mimic clinical settings. Otherwise, promising preclinical studies that even go into phase 2 trials would have a setback and will not move to the next phase. At this phase, at this juncture particularly, I strongly believe that cell therapies are moving beyond the innovation phase and will need a well-oiled orchestration phase where the conduct of initial preclinical studies will be of utmost importance. The next one is the capability of engineering cells. We know that tumor microenvironment is very complex, and particularly in highly malignant GBMs, we need to target multiple components of the tumor microenvironment for making cell therapies effective. As such, we'll need engineering cell types that can target multiple antigens and release immune modulators and molecules targeting immune checkpoint proteins. Therefore, we need a cell type that can be heavily engineered and fits the bill. This will define the choice of cell type we can use for a particular tumor. We know there's complexity of engineering cells. I envision a combination of different cell therapies to treat highly malignant tumors, particularly GBMs. One cell providing the payload and the other targeting multiple antigens. And this leads to my final, sort of bottleneck or choice that we have to make is the choice between the allogenic versus the autologous autologous therapies and the allogenic off-the-shelf therapies. In many solid tumors, particularly GBMs, time between diagnosis and surgery is very limited, and that's where the transplantation of the cells will happen. So there's not enough time between the diagnosis and the and the transplantation of cells into the patients. And in that case, the choice of cell type to be used has to be allogenic, as opposed to the autologous, which is mostly useful in recurrent settings. So in all, I think these are basically the bottlenecks and the choices we'll have to make as we move these cellular therapies into highly malignant tumors.
Great. Thanks so much, Khalid, and best of luck with your work.
Thank you.
Number 11, Antisense Oligonucleotides, a novel therapeutic platform for neurodegenerative disease.
Some gene therapies seek to repair or replace what's been lost, like genes that are abnormally silent because of a genetic misspelling that terminates their usual function. But what happens when genes are broken in a different way that gives them new, often unexpected behaviors? Scientists have devised innovative technologies to neutralize such wayward genes. One involves a class of gene therapies known as Antisense Oligonucleotides, or ASOs, which consist of short fragments of DNA, or sometimes its chemical sibling, RNA. These fragments are synthesized in the laboratory to match the genetic code of the gene of interest. Such precision allows the activity of the target gene to be shut down at its molecular roots. Importantly, ASOs are relatively straightforward to engineer, so they can often be designed more quickly than other therapies. Now, the field of ASO-based therapies is rapidly expanding. Over the last 4 years, 6 new ASO drugs were approved by the FDA, and many more are under development for a range of conditions, including neurodegenerative diseases such as ALS, Huntington's disease, and Alzheimer's disease. There is a desperate need for drugs that can significantly slow or even stop the progression of these devastating conditions. For example, clinical trials of an ASO drug that silences a gene involved in some forms of ALS are now underway. The results are encouraging and point to the promise of this therapeutic approach for treating patients with neurodegenerative diseases.
Hi, Merit. Welcome. This field is at a pivot stage clinically. Can you comment on where we are clinically with these types of therapies and what's next?
You're absolutely right. We're at this amazing time in ASO treatments for ALS and other neurological illnesses. For ALS, we now have 3 active clinical trials for with different targets with ASOs in people. One of them, called Tofersen, is in late-stage phase 3 testing. We hope to have the results soon, in a couple months. And if that's positive, we could see that coming to patients even this year. That is a particularly brutal form of ALS where lifespan can be less than a year. So we're all eagerly waiting for that. There's also planning for a prevention trial for that same genetic form using that same ASO. And that's completely new in ALS to try to intervene before the first symptom. But we're also using ASOs in ALS for sporadic disease, targeting proteins of interest like ataxin-2 or staphymin. So we're moving those treatments also into trials for the broader form of ALS. We're very excited about this approach. And we're seeing it, as you mentioned, in Huntington's disease, in Alzheimer's disease, and other illnesses where we have well-defined targets that we can modify with ASO approaches.
Well, thanks so much for all you do, and congratulations on the D12 nomination.
Number 10, improving gene therapy through designer viruses.
Viruses found in nature have become essential vehicles for gene therapy. One virus, known as adeno-associated virus, or AAV, is a popular choice among gene therapy developers because of its long track record of safety and efficiency in animal studies compared with other gene delivery systems. But AAV is not a perfect solution. The virus is not always efficient at reaching particular organs and tissues in humans. Therefore, higher doses must be used to produce a health benefit, and That can lead to toxic side effects. In addition, naturally occurring AAVs have affinity for a relatively narrow set of human cells and tissues, limiting the diseases that can be treated. Also, since AAVs are relatively abundant in nature, many people have been exposed to them. As a result, the individual's immune systems are often already armed against AAV, which could blunt the therapy's effectiveness and result in severe immune responses. Scientists are creating designer AAVs in the laboratory to help overcome these challenges. Using data-driven methods, they are modifying the molecular makeup of the viruses to generate enhanced versions that home to specific organs, like the lung and kidney, which are not targeted by the current slate of therapeutic AAVs. Researchers are also fine-tuning AAVs to infect some cells in a tissue, but not others. For example, a specific subtype of neurons in the brain. Finally, efforts are underway to create AAVs that can evade detection by the immune system, which would help expand the clinical impact of gene therapy by making more patients eligible to receive it.
We have Dr. Casey McGuire, one of the MGB scientists who has been at the forefront in engineering better ways of delivering AAVs and better AAVs. Casey, what excites you the most about this technology?
Thanks a lot, Nino. Yeah, it's a very exciting time to be in AAV-based gene therapy. Despite the technology being developed for about 40 years now, I'm just excited that we haven't really seen the ceiling for the technology. So AAV's been really amenable to a lot of different modifications. Like we ran into the problem with the packaging capacity being too small. So people engineered the trans-splicing vectors. We had a problem with efficiency, and now there's AAV9 variant capsids that are about 1,000-fold more efficient to the crossing the blood-brain barrier compared to the clinical standard AAV9. And now we have some issues in the clinic with immune responses with AAV, in some cases, not all. And there's some really promising strategies out there, which I'm sure different groups will solve. So I think this speaks as much to the pioneers and the new people working in the field as much to the technology, but really, I don't think we've reached the ceiling yet of the technology. So thanks.
Thank you, Casey, and good luck with your work. We're really looking forward to what you will come up with.
Thanks, Nino.
Number 9, building the next wave of CAR T cell therapies.
In 2017, the FDA approved the first CAR T therapy, a groundbreaking form of gene and cell therapy in which a patient's own immune cells are isolated, genetically engineered in the laboratory to endow them with certain therapeutic properties, and then infused back into the bloodstream. For difficult-to-treat blood cancers, CAR T therapies have proven to be remarkably effective, with some patients living for years cancer-free. Now, researchers are working to expand the impact of this technology. In the current paradigm, the therapies are custom-made for each patient through a complex procedure, which not only drives up cost, but also limits the number of patients who can access them. But new approaches could help streamline the CAR T process, including advanced cell therapy manufacturing platforms designed for scale and automation. Efforts are also underway to broaden the diseases that CAR T therapies can treat. Those include the development of CAR T therapies that can kill solid tumors. They also include the design of novel CAR T therapies that target entirely new areas, like autoimmune disease. It may also be possible one day for CAR T therapies to be used off the shelf, which means patients would not need to supply their own cells as a starting material. Instead, doctors would select from an assortment of pre-made options. Although the development of universal CAR T therapies is in its infancy, it could help expand the clinical utility of these innovative medicines.
It is my great pleasure to introduce to you Dr. Marcella Maus, who is one of our MGB scientist clinicians. She's one of the superstars in the CAR T cell technology field. Marcella, what areas of innovation do you think are going to be really critical and instrumental in advancing CAR T cell technologies to more patients?
Thank you so much, Nino. I think what the whole field is really waiting for is some proof of concept that CAR T can be engineered to have some clinical efficacy in solid tumor. I think there are lots of new transgenes being tested, dual targeted CAR Ts and CAR Ts that are armored in some way. And so I think once we start to see proof of concept in solid tumors, that's really going to be the next release, the next wave of innovation. And then I think that the two main areas of innovation that follow would be to really improve the manufacturing process, to have all closed systems that are very rapid and scalable and automated. I think right now the manufacturing process is the least scalable portion of generating CAR T, and so having new ways of doing that will really unleash the potential of CAR T. And finally, I think like the whole field of gene therapy, new ways of gene transfer are going to be really important. Right now, we use mostly viral vectors, which are incredibly efficient in terms of generating transduced T cells and even transduced immune cells that are not T cells, such as NK cells or iPS-derived cells and various families of T cells. But making viral vectors is quite expensive and requires a lot of PhDs and clean room facilities. I think if we can start to have a more scalable and closed system and automated manufacturing of vectors or alternative gene transfer mechanisms that are as efficient as viral vectors, but that also maintain sort of fidelity to the genome, those are really going to be important for the field to move forward.
Thank you, Marcella. We're looking forward to your work.
Thank you.
Number 8, replacing what's lost. Stem cell therapies for diabetes.
Over 1 million people in the U.S. live with type 1 diabetes, a chronic disease in which the immune system destroys insulin-producing cells in the pancreas. Patients can often keep the condition in check by monitoring their blood sugar levels around the clock and injecting themselves with insulin multiple times daily, or wearing a small pump that allows delivery of insulin under the skin. But all of the available treatment strategies are labor-intensive and imperfect, and mistakes can be life-threatening. Now, a new approach is emerging that involves replacing the lost insulin-producing cells, called beta cells, with a special laboratory-grown variety. Over the last several years, scientists have developed a few recipes for generating these cells, using different stem cells as the key ingredient. But these approaches all share the goal of producing sufficiently large numbers of cells to meet patients' needs. There is another important requirement. The replacement cells must be protected from the immune system. Otherwise, they'll be destroyed once inside the body. Researchers have devised a handful of cloaking strategies, including specialized gels and implantable devices. Efforts are also underway to enable replacement cells to release their own immune-blocking signals and help shield themselves. As these technologies continue to advance toward the clinic, researchers hope to bring them to bear on another disease, type 2 diabetes. Worldwide, there are over 400 million people with type 2 diabetes. Many require insulin injections, underscoring the need for a more durable solution.
And we have here with us Dr. Mary McDonnell. Dr. McDonnell is the director of the Brigham Diabetes Program. Mary, as a world leader on diabetes care and science, when do you think these stem cell therapies will be available to patients?
It's such a great question, and it's a dangerous one to answer. And I feel like I represent the patients and all the clinicians around the world waiting with bated breath for this. But it looks like Vertex's product will be first in line. This is a stem cell-derived, fully differentiated cell product that does require immunosuppression to prevent graft rejection. And as we heard yesterday, that's not the ultimate goal, but it probably will be the first product available to patients. They're close to moving into phase 2 with this product. So with the primary completion target of 2024, one might add another 3 to 4 years for the phase 3 study. So we're looking after 2027 and hopefully before the end of this decade. And it really does look like we're on track. Now, ViaCyte has
a somewhat similar trial, but there are perhaps more details they need to work out because they are moving toward co-developing the encapsulation device and really working toward a cloaked, if you will, cell product to avoid immunosuppression. So if the first product will be the encapsulated immune engineer, the timeline will be longer. No one really is in that clinical space except ViaCyte. So they do have a trial that's testing those cells, and early data was a bit disappointing, but this has only inspired the company to work harder, and they are really gearing up, restarting the trial with a better device, from my understanding. So if everything goes super well, we should see a product be in the clinic before 2030.
Great. Well, thanks so much, Dr. McDonald. Over to the next one.
Thanks a lot.
Number 7, from skin to neurons, a groundbreaking cell therapy for Parkinson's disease.
Parkinson's disease affects some 10 million people worldwide. Although the precise cause remains unclear, a mix of genes and environment are thought to be at play. Regardless of the underlying trigger, patients with Parkinson's disease suffer from a progressive loss of neurons in the brain that deploy dopamine, an important signal for controlling movement. Unfortunately, there is no drug available to protect or stop these neurons from dying, although researchers have worked for years to find ways of replacing them. Now, enterprising teams of scientists and clinicians are making progress towards this long-sought goal. One method goes something like this. Isolate the immune cells from a patient's blood sample. Bathe the cells in a cocktail of specialized molecules in the laboratory to coax them to become dopamine-producing neurons, and surgically implant them in the brain to replace the lost neurons. Although this cell therapy does not fix the root cause of Parkinson's disease, it could provide a functional cure by replacing the dopamine-producing neurons in patients' brains and restoring normal movement to their bodies. This approach, while innovative, is also complex and costly because replacement cells must be custom-made for each patient. However, there is also an important advantage. Since the cells are derived from patients' own bodies, there is no need for immunosuppressive drugs, as is typically required when foreign cells and tissues are transplanted.
And we have here Dr. Ole Isaacson, who's a professor of neuroscience at McLean Hospital. Welcome, Dr. Isaacson. In your experienced view working on these diseases, what is the expectations from a patient's perspective, what this first generation of therapies could mean for them?
Thank you, Luke. Yeah, so this is the treatment that specifically replaces the patient's own dopamine cells, which are lost in the disease process and creates the characteristic movement disorder. And so it's the only one that really can get rid of all the dopamine drugs. And these cells, once transplanted, actually can restore normal neurotransmission. And by that, we mean release and uptake of dopamine, which is what the real problems is with the current treatment for the patients, because the problems of dyskinesia, extra movements with all dopamine treatments, and then the wearing off of these treatments. So eventually, we believe this cell therapy, if shown safe and effective, could become a first-line treatment in this for this disease, not only by transplanting dopamine neurons, but probably and possibly in the future also some of the support cells that are part of the disease process. So I feel that this regenerative medicine for patients, personalized and replacing chemicals that are used throughout the body, is disruptive in many ways, as we've heard from others. It's disruptive as a modality. Autologous cell therapy actually in our model suggests that they work better than allogeneic, because nerve cells actually use a number of their own proteins to guide axons. And actually several studies indicate that the autologous are the better one. Also, it's transformative because it's local, and we place the neurons back in a system which is now functionally curing the patients.
Very good. Well, thank you so much.
Thank you.
Number 6, eyes and ears, expanding gene therapy's reach.
Among the very first gene therapies to be approved in the U.S. is one aimed at a rare form of blindness, caused by a disabled gene named RPE65. Using highly specialized equipment, doctors inject the one-time therapy directly into the eye, delivering a virus that carries a healthy copy of RPE65 and helps restore vision. This remarkable success has led to a wave of other gene therapies for the eye, which are now under development. Some 200 genes in humans are directly linked to vision problems, underscoring the incredible potential of the technology. In addition, the eye is well suited for gene therapy. It's readily accessible to treatment. It's among a handful of sites in the body where the immune system is less active, and it's largely sealed off, so any injected medicines are unlikely to spread to other areas. Scientists are also pursuing novel gene therapies for another sensory organ, the ear. Millions of people in the U.S. suffer from hearing loss, yet there are no FDA-approved medicines to treat it. Over 150 genes are tied to hearing loss and deafness. Several gene therapies are now being developed that carry healthy versions of some of these genes into the inner ear. Unlike the eye, the inner ear is difficult to reach with therapeutics. To help overcome this hurdle, scientists have fine-tuned the molecular makeup of the viruses used in gene therapy to create versions that can penetrate the ear's internal structures.
Joining us here is Dr. Patrizia Musolino, who's a neurologist and scientist at the Mass General Brigham. Patrizia, when you look at this field, what do you think are some of the big obstacles that could jeopardize the future use of gene therapy for patients?
Thank you for having us and for the question. There are multiple challenges ahead. The ability to now do gene therapy and do it with successful trials in the eye and the ear, which are compartments, and they're easy to access and they have the immune privilege, really accelerates the evidence of what are our challenges coming forward. And I think disease identification in early diagnosis is going to be one of the biggest challenges, expanding prenatal screenings and newborn screening to be able to get to the patients in asymptomatic stages. And that changes in the regulatory aspect. What is the endpoint and what are the biomarkers that we are now aiming to change when we're talking about a one and done and a permanent gene change? And that will expand the timing of interventions for more patients that are affected by the disease. And as we were seeing in the forum, the discussion about what's meaningful to the patient, the endpoints may not be what we think is the right endpoint. We really have to hinge on what the patient's quality of life and ability to enjoy life is and what's meaningful to them. And I think it's still a challenge to identify the critical cells and genes that we need to correct. Not everything matters and is as critical in the disease. And that takes us to engineer better delivery systems, AAV-based, non-viral nanoparticles to improve the efficiency, the safety, manufacturing capacity, and the cost. And lowering the cost and improving the access to this therapy is, I think, the biggest challenge we have right now to make it worldwide accessible and equitable as a therapy.
Thank you for your thoughtful answer.
Number 5, a faster way to mobilize bone marrow stem cells.
Bone marrow stem cells are critical for treating disease, but they are not easily accessible. To donate bone marrow stem cells, either for themselves or for others, patients must first receive injections of a drug that coaxes the stem cells out of the bone marrow and into the bloodstream. Summoning a sufficient number of stem cells requires daily injections over several days, which can cause pain, nausea, and other complications. Then, the cells are harvested from the blood in an hours-long collection process. What if there were a simpler approach? Scientists are now testing a potential alternative. It involves a single injection of a drug that mobilizes bone marrow stem cells very rapidly, reducing a 5-day protocol to a few hours. This new method holds promise, not only for bone marrow transplantation, but also for gene therapies that depend on manipulating bone marrow stem cells. These treatments, known broadly as ex vivo gene therapy, require isolating bone marrow stem cells, treating the cells outside of the body with gene therapy, and then infusing the modified cells back into patients' bloodstream. Currently, one ex vivo gene therapy is available in the US for patients with a rare genetic disease. As the pipeline of ex vivo gene therapy expands to include treatments for more common diseases, this new approach could help streamline the isolation of bone marrow stem cells and ensure that this important step does not become a stumbling block.
Joining us is Dr. Jonathan Haggett, who is one of the researchers at Mass General Brigham who works on this problem. Jonathan, thank you for joining us. What do you think is the greatest potential for this innovation in improving patient care?
Thank you, Nino, for the question. I think there's three immediate patient impacts that come to mind. So for allogeneic transplant, where you find a healthy donor who is a compatible match for a patient, it's often difficult to find a suitable donor, particularly for minority groups. So anyone listening, please sign up for the bone marrow registry if you have not already done so. But currently, of those people who have already done the cheek swab and are on the registry, 1/2 decline to donate. And this is for many reasons, but a significant reason is the inconvenience, the side effects, and ultimately fear of the current standard of care. So this disruptive technology is one injection, and an hour or so later, you donate stem cells connected to a machine, very similar to donating platelets or plasma. So this technology has the potential to make the process of donation dramatically better for the donor, and ultimately could lead to more available donors for patients in need. I think secondly, a key hurdle for ex vivo gene therapies, many of which have been discussed in this forum, is the ability to collect high numbers of quality stem cells. So this technology so far has demonstrated that not only is it much faster than the current standard of care, but the quality of the stem cell harvest is also better. So higher quality stem cells at the beginning of the gene therapy process is likely to make that more efficacious. And finally, related to the gene therapies, some patient groups are not able to tolerate the current standard of care for collecting stem cells, such as those with sickle cell anemia. So based on the rapid nature of this disruptive technology and its deferring mechanism from the current standard, the ability to collect stem cells from sickle cell patients for gene therapy may also be significantly improved.
Thank you.
Number 4, moving beyond viruses to fairy genes.
When genes are used as medicines, they must gain entry to cells in the body to do their work. From a biological perspective, that's a complicated task. Decades ago, scientists recognized that nature had already invented a solution: viruses, which are essentially miniature syringes filled with genes. The first wave of gene therapies to reach the clinic use highly customized viruses, which have been molecularly honed and tailored to allow for the safe, effective delivery of human genes. Now, as scientists seek to build the next generation of gene therapies, they are pursuing new options for gene delivery. One reason? Viruses are limited in how much genetic material they can carry, which means large genes cannot be accommodated. Also, if gene therapies can be targeted more precisely to specific organs or tissues, they could be used to treat a broader range of disorders. And because the human immune system has evolved to recognize and neutralize viruses, it can be a formidable obstacle to effective gene therapy. Nanoparticles are one area of active research. Using inexpensive nanoscale materials, researchers are creating highly sophisticated beads that help protect and direct gene therapies to their intended destination within the body. A variety of materials can be used to construct nanoparticles for gene therapy that differ in molecular composition, size, and electrical charge, which helps control when and where therapies are released in the body, thereby reducing toxicity. These efforts have been boosted by the recent development of a pair of highly effective coronavirus vaccines that use lipid-based nanoparticles to deliver their therapeutic cargo.
With us here is Dr. Natalie Artsy. Natalie is faculty at the Brigham and has really devoted her career on progressing the science of non-viral gene transfer and nanoparticles. Natalie, from your experience and given the revolution that we've gone through over the past year with nanoparticles and coronavirus vaccines, tell us, what does this mean for the field of gene therapies, and how has this propelled your field?
Thank you, Luke. So I agree. The remarkable success of COVID-19 really exemplifies some of the founding principles behind nanomedicine and the ideas of drug delivery, which is that the rationally engineered materials, thanks to their nanosize and physicochemical characteristics, can protect drug cargoes from degradation, offer control over their biodistribution, intracellular localization, and release. The approved mRNA vaccines showed safety and really unprecedented efficacy that will also serve probably as a springboard for new applications of nanomedicines. Until the COVID-19 crisis, nanocarriers have been really been explored for oncology primarily. Now we can think about oncology treatment more broadly and even consider personalized therapies, since we can develop mRNAs relatively rapidly to provide and form with tumor antigens that can help the body recognize the tumor and attack it. Another area is immunizations for infectious diseases like tuberculosis, HIV, and flu. We can think about mRNAs that encode multiple flu strains to provide with better protection. The last year also demonstrated the speed and flexibility by which vaccines can be designed and developed, and can perhaps be used now for rare diseases, rare genetic diseases, where the population of a patient is smaller. And finally, nanoparticles can help materialize other types of gene therapy, like DNA gene therapies, CRISPR gene editing, and epigenome editing. Now that it has been revealed what can be accomplished under the right circumstances, the continued collaboration and partnership between industry, academia, regulatory agencies, and healthcare systems is really critical to the continued development of unprecedented therapies that could not have been imagined before.
Thank you so much, Dr. Artsy.
Thank you.
Number 3, all about that base. The rise of a new class of genome editing tools.
Technologies that make specific changes to the genome, a capability known as genome editing, are accelerating the field of gene therapy. A popular genome editing technology called CRISPR-Cas9 took the scientific world by storm several years ago with its precision and ease of use. Researchers have since discovered that the tools can make various errors during the editing process, in part because they work by cutting both strands of the DNA double helix. Those qualities are not a good match when it comes to correcting an important class of genetic mutations known as point mutations. These mutations cause disease by changing a single base of the genetic code, like altering just one letter within an entire book of words. Such changes can be inconsequential, but some can be harmful. More than 30,000 point mutations in the human genome are known to cause disease. With the recent emergence of an exciting new class of genome editing tools, known as base editors, it is now feasible to correct these point mutations. By making nicks in the double helix, cutting just one strand, and recruiting a special enzyme to these sites, base editors can fix single-letter mistakes. The first gene therapies that harness these new base editors are now in development for a range of disorders, including sickle cell disease, inherited blindness, and genetic forms of high cholesterol. As base editing technologies continue to mature, researchers are also working to apply them to more common diseases, such as Alzheimer's disease and cancer.
So here at MGB, we have the privilege to be surrounded by some of the leading experts in this field. And with me here is Ben Kleinsteiver. Ben is faculty at the MGA Center for Genomic Medicine. So Ben, especially also with some of the recent news, where do you believe that base editing technologies will have the greatest therapeutic impact?
Thanks, Luke. Your question about the clinical impact of base editing is really important and timely. Backing up a bit first, base editors allow researchers to make precise changes to the genomes of living cells and organisms. So to answer your question, there are a few main factors that dictate how broadly useful base editors will be in the clinic. First, the genetic mutation that caused the disease is highly important, since base editors permit us to make a subset of all possible single-letter DNA changes. Immediately, this means that base editors will be most useful for treating monogenic diseases caused by well-defined mutations. Recently, studies have shown the potential of base editors to treat various monogenic diseases, including sickle cell disease, deafness, blindness disorders, progeria, and many others. Base editors also allow us to knock out genes and their expression, making them amenable to therapies for more complex diseases like cancer and cardiovascular disease. A second major consideration for the impact of therapeutic base editing is the tissue or organ that needs to be corrected and treated. Certain diseases can be corrected ex vivo by extracting cells from a patient, editing them in a dish, and reinfusing them to treat disease. However, for treating most diseases, that will require in vivo editing to correct a large fraction of cells and tissues or organs. In vivo base editing will benefit from a lot of what you've heard about already today, tissue-specific viruses or nanoparticles, for which we've heard lots of exciting progress today and in the field. So given these general parameters, in just a few short years, there's been a lot of really exciting progress developing base editors designed to treat both rare and common diseases. With base editors, it's now possible to make very specific changes to the genomes of patients while achieving safe, efficient, and durable editing without obvious genotoxic consequences. So I think the prospect of long-lasting one-and-done cures with base editors holds tremendous promise to transform medicine in the coming years.
Great. Well, thank you so much, Ben.
Number 2, a genetic fix for 2 common blood disorders.
Blood cells depend on a critical protein called hemoglobin to ferry oxygen throughout the body. Genetic mutations can cause the production of abnormally structured hemoglobin or reduce the amount of this protein, causing devastating diseases such as sickle cell disease and beta thalassemia. These conditions are common and difficult to treat. The only cure is a bone marrow transplant, which is risky and often not feasible due to the lack of a suitable donor. Yet, new genome editors, tools that make precise changes to a person's DNA, are paving the way toward a different kind of cure. One approach uses a type of genome editing called CRISPR and involves some creative genetic tinkering. Instead of repairing the damaged hemoglobin gene, CRISPR can help reactivate another gene, the one for fetal hemoglobin. Typically, this form of hemoglobin is made early in life, but is deactivated as adult forms are made. However, when the fetal hemoglobin gene is switched back on in blood cells of patients with sickle cell disease and beta thalassemia, it can substitute for the missing or faulty adult version. This CRISPR-based gene therapy is now being tested in clinical trials, and early results are encouraging. Other gene therapies are also in the works, including those based on older technologies that augment rather than repair defective genes. While all of these new therapies are promising, they will be expensive. That means further innovation is needed to drive down costs, especially for patients without access to state-of-the-art healthcare.
David, how are you? We're fortunate this morning to be joined by David Skadden, a leader in this area. David, given your leadership role in this particular field of blood disorders and gene and cell therapy, if you can give us a sense of access, how do we give access to those patients who need these therapies the most?
Thanks, Robbie. Well, I have to say, it's one of the great things is that we finally have something that can really make a difference for these people. What we know is that these approaches really do change the lives of people who are able to get them. As you point out, the problem has been that very few people have been able to, in part because of the newness of the technology, but also because of the difficulty that people go through to get the therapy and its cost. So there are a couple of things that we can do to at least improve the experience of those people. And one is that we can make it easier to acquire the stem cells. And that's something that you just recently heard about from John Haggett with the technology that he developed. The other is that when people have their stem cells modified, in order for those to be able to come back in and establish themselves in a person, we have to give them therapies that eliminate the existing stem cells. And that means exposing them to very toxic chemotherapy. So we have to be able to make that something that doesn't have all the collateral damage of that kind of therapy. And that is an area of active investigation. I'm involved in that, but others are as well. And we hope that that will then make it so that this approach can be something that could be done as an outpatient. The final piece of this, though, will still be to be able to do it so a person doesn't need a transplant at all, but we could give some kind of therapy that would be just administered intravenously and allow it to work on the existing cells. That, I think, is still a long ways off, but we're getting there. And ultimately, we hope that we'll be able to also have cells that don't require each individual to provide their own stem cells, but to have something that can become an off-the-shelf source. That's even further out, but I think we'll get there. The good news is that we're making gradual, but very significant headway in making it so that these people who've otherwise suffered so terribly have an opportunity to go back to living a normal life.
David, thank you very much, and thank you for your leadership in this area.
Thank you, Riley.
Number 1, a new generation of cholesterol-lowering therapies.
The health risks of high cholesterol, especially for the heart, have been clear for decades. That's why doctors advise patients to keep their blood cholesterol levels in check by adopting healthy habits and, if needed, take a cholesterol-lowering drug. But those drugs don't work for everyone. Scientists have been working to devise new treatments for high cholesterol, as well as other harmful fats, called lipids, that circulate in the bloodstream. Over the last several years, researchers pinpointed key genes involved in cholesterol and lipid metabolism that represent promising targets for new cholesterol-lowering treatments. Now, a wave of groundbreaking gene-based therapies is on the horizon. One of these new therapies works by silencing a critical gene called PCSK9. Instead of disabling a disease-related protein, as most conventional drugs do, gene silencing therapies prevent the protein from being made at all. That durability means patients could receive an injection of gene silencing drug every 6 months to control their blood cholesterol. Another transformative genetic medicine is also under development. It packs the power and precision of a word processor and can alter the instructions written in a particular gene by just a single letter. The promise of this technology, known as CRISPR base editing, lies both in its precision and its potential permanence. Patients may be able to undergo a one-time treatment and maintain healthy cholesterol levels for a lifetime.
Good morning, Cricket. We're incredibly fortunate at Mass General Brigham to have world leaders in the gene and cell therapy space. And of course, synonymous with gene and cell therapy and cardiac disease is Cricket Seidman from the Brigham. Cricket, in this particular area, in this crowded space of other lipid-lowering therapies, how do gene and cell therapies fit in this particular landscape?
I think they have enormous potential. And increasingly, we are seeing that it is not only potential, but it is soon going to be a reality. We know that many people, despite the many lipid-lowering drugs, continue not only to have elevated cholesterol, but more importantly, to have adverse clinical outcomes associated from that, including heart attack, stroke, and vascular disorders. And we now have an opportunity to really treat them effectively and permanently, either by Cas-CRISPR silencing of PCSK9 or base editing. Indeed, a recent publication that came out this very week showed the efficacy of reducing cholesterol levels and LDL in particular levels in animal models, large mammals that in fact have had editing of their PCSK9 gene. And not only has it been therapeutic in terms of reducing lipids, but this kind of editing can occur without significant deleterious effects in other genes that are encoded within the liver cells that make PCSK9, or indeed in cells around the body. So it really is an enormous opportunity, and I think we will see this as an early winner of gene and cell therapy for treating a very common disorder, atherosclerosis, the #1 cause of heart disease and cardiac death around the world.
Really exciting, Cricket. Thank you very much for your leadership in this area.
Thank you.
So that brings us to the end of the Disruptive Dozen. I suspect Chris Coburn will ask us to have a baker's dozen next year, just given the remarkable number of innovations and disruptive activities going on at Mass General Brigham. So really terrific session, and thank you to the investigators and the co-chairs that we have here. Let me turn it now to Nino for some closing remarks.
Yes, like all good things, things have to come to an end. And this has been a remarkable 3 days on gene and cell therapy. Really wanted to thank the distinguished panel members and all the presenters for joining us throughout this year's forum to share their perspective and expertise in this field. We are deeply grateful to have such an accomplished group offer their expertise and their insights on the potential of gene and cell therapy. But most of all, we also want to thank you, the thousands of attendees that joined us online. Registration for this year's World Medical Innovation Forum has shown the urgency in advancing gene and cell therapy to the front lines of medicine so that we can benefit patients everywhere.
Well, I will argue with Nino a little bit that all good things don't come to an end, because as Chris said, we have another forum next year. That said, we've heard about this vital role during the meeting over the last few days of collaboration, and you've heard collaboration among academics and industry and academia, regulation and regulators and so forth, really just remarkable. Our goal, of course, is to address rare disease, ultra-rare disease, and even common diseases, and you've certainly heard that. But in that context, ensuring equity and access to patients is unbelievably critical, and certainly a mission of Mass General Brigham. We've heard how gene and cell therapy is at a commercialization inflection, and the last few days undoubtedly would leave you with that impression. Moving forward, we're certainly committed to ensuring that patients remain at the center of this process. As we heard even just in the last session, from the very beginning of the journey, patients are at the center. And with that, we will continue to address equity, safety, cost, manufacturing efficiency, the regulatory framework, among the many complexities. Luke?
Yeah, I think the question is, how do we get there? How do we get to patients? How do we solve all these challenges? And I think we've heard also over the past couple of days that the continued need is there to have a pipeline of invention, a pipeline of investment, a continued emphasis on the identification, development, and delivery of safe and effective new tools and treatments. Throughout the meeting, I think we all sensed great optimism regarding the power and the versatility of cell and gene therapy breakthroughs. But again, most important, we heard of the day-to-day impact that these therapies, these cell and gene therapies, can have on patients' life. And it's really up to us. And us here means the academic world, the industry, venture, the regulators that were here at the forum, to continue to discover, invent, invest, and importantly, expand the access to these unique therapies. Maybe as Jack Hogan, who's a young patient at Mass Pioneer, who was treated here at Mass Pioneer with Luxturna, one of the MGB hospitals, he shared with us on the first day in one of the first panels of the forum something when he was thanking the PI of the study, or actually the clinician who treated him. Without you, miracles like me will never happen.
So throughout this forum, we have been polling the audience with a number of questions, and we would like to share the results of this. So the first was related to expansion of gene and cell therapy will be gated mostly by. And as you can see the answers here, it is interesting that we still don't know which one specifically, but we do think that we need all of these responses, safety, better preclinical developmental models, avoidance of serious side effects, and occurring delivery and targeting challenges. The next slide had to do with the valuation in the gene therapy market space, and we'll is this going to remain stable or grow in the next 3 or 5 years? And there's tremendous optimism here. Over 68% of you agree with this statement, that there is going to be continued growth in the valuations in this space. Next slide.
Thank you, Nino. We had two other questions that all of you participated in. The biggest challenge in making GCT more widely available to patients. It's pretty impressive that 54% chose cost, reimbursement, and access. And we heard about issues of these critical matters and how they affect our patients from David Skadden and Patricia Mussolino and others. So not surprising and unbelievably important for us to address if we need to move this field forward. Thank you. We'll go to the next slide. The last question we have is, I believe that GCT will emerge as a widespread game-changing therapy and treatment. And here, the majority of people, 44%, said in 5 to 10 years. It's interesting that the first monoclonal antibody came to us in 1986, and only several years later did the next one and the next one come out. Obviously, you've heard during this meeting the pace of and the accelerated pace, I should say, of the first and then the second and the third here. So I suspect 5 to 10 years is certainly mildly conservative, but certainly we're optimistic about that. Let me now turn it back to Luke for the final comments. Luke?
Thank you, Ravi, and thank you, Nino. It's my honor to kind of close off here. And as Ravi mentioned earlier, we look forward very much to reconvene here in person May 2nd to May 4th, 2022, for the next installment of this forum. We'll continue there the dialogue on cell and gene therapy and the important work that lies ahead. I thank you all for attending, and I wish you all good health and look forward to seeing you here in Boston in person next year.